Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about protein structure and protein folding. So what we're going to see here now that we have a good handle on amino acid structure and function and how peptide bonds are formed between amino acids to build a polypeptide, we're going to start getting into the step-by-step manner in which proteins fold into a final three-dimensional structure. It all starts with what is called the primary structure of the protein, which we'll talk about in just a minute, but is basically just the linearized sequence of amino acids in the protein. The protein will then fold into localized secondary structures. Finally, far-away portions of the protein will come together to form what is called a tertiary structure, and these three structures together make up the folding sequence of a single polypeptide, just one single contiguous stretch of amino acids within a single polypeptide. There is a fourth level of protein folding called quaternary structure, but this only applies to multiple polypeptides that assemble together and form a multi-polypeptide, multi-subunit protein. So the primary structure refers to the linear sequence of amino acids in order from N-terminus to C-terminus. So this primary structure is going to give us three very important pieces of information about any particular protein. Number one, it tells us the number of amino acids that are in the protein. Some proteins are very short, some proteins are very long. How long or how short a protein is dictated by how many amino acids are joined together by peptide bonds. Next, it tells us which amino acids are in the protein. Remember, there are 20 different amino acids that are used to build proteins. Some proteins will contain all 20 of the amino acids. Some proteins will be missing some amino acids. Amino acids can be repeated as many times as we want. Amino acids can be excluded. It just depends on which protein you're talking about. And finally, it tells us, most importantly, in which order are those amino acids found. Is the first amino acid methionine? Is the last amino acid valine? That, again, is going to depend on which protein we are talking about. So to visualize the primary structure for a protein, you can picture the fully built polypeptide with all the peptide bonds in place as being kind of like a string of beads that is stretched out in linear fashion. The amino acids are found in the order that is dictated by the genetic code, which the cell and all of the cell machinery will use to interpret the instructions that are found in the genes on the DNA and the instructions that are found in the mRNA copy that is made after transcription. You can see here that this primary sequence of this polypeptide has the N-terminus here and the C-terminus here, as we said we would in the previous video. So what we're going to see here is that the first amino acid that is added by the ribosome is found at the N-terminus, and the most recently added or last amino acid that was added by the ribosome is found at the C-terminus. In a future video, we will emphasize this point even more, but what this tells us is that proteins are built in the direction from the N-terminus to the C-terminus. So whichever amino acid is at the N-terminus, that was the first amino acid that was added and the first amino acid that was found in the genetic code. So the primary sequence of a protein is represented, as we saw in the previous video, by using one-letter codes for each of the amino acids. So you can see the first amino acid that is found here at the N-terminus is methionine, then aspartate, glycine, leucine, serine, asparagine, arginine, valine, aspartate, and then finally lysine. So this is a very useful way of representing all of the amino acids that are found in a protein, especially when that protein is very long. So one last important point about primary structure is that even though we do not currently have a good and reliable way of predicting a protein's folded shape or a protein's function just based on the primary sequence alone, we do know that the primary sequence of a protein is all that is necessary to determine the shape and the function, even if we don't really have a good way of predicting that. So what that's really telling us is that which amino acids we find in a protein and in which order they are found is extremely important in determining what that protein is eventually going to do in terms of cellular and physiological function. If we were to change out amino acids or swap the order in which they are found, we would totally and irrevocably change the function of the protein, probably for the worse. Okay, so now we are going to start moving on to secondary structures. Before we get there, though, we need to talk a little bit about how proteins are actually going to fold in a practical way. So when a protein begins to fold, the linearized polypeptide, which we just looked at in the primary structure, is going to need to bend and twist into a discernible shape. The problem is that the peptide bonds, which are being pointed at here with blue arrows, are inflexible because they have partial double bond character. There's not much rotation that can go on there. So if we're going to bend and twist the protein, it cannot be done at the peptide bond level. These other bonds here, which are called phi and psi bonds, which are highlighted here in orange, these are not the peptide bonds, and since they do not have double bond character, they have full 360-degree rotational freedom. So we can rotate the functional groups in the atoms about these axes in all 360 degrees. So when these bonds rotate about their axis, the polypeptide can and will be bent into a variety of different shapes. So moving on to secondary structure, these are localized shapes within the polypeptide that are formed as a result of the chemical attractions between amino acids that are usually pretty close to each other, usually less than 10 positions away from one another. So a couple of other important points. These secondary structure interactions will not involve the amino acid side chains, so secondary structure interactions are always going to happen between atoms that are in the backbone rather than in the side chains. And secondary structures are held almost entirely together by hydrogen bonding, which we have obviously talked about before. To that point, there are two major secondary structures that are very commonly found in a lot of fully folded proteins: alpha helices and beta sheets. So if we look at an alpha helix, the word helix here should be fairly familiar to you in terms of visualizing what it looks like. This is going to produce kind of an ascending staircase-like structure here. The backbone twists around itself and upwards. So what this is going to allow for is hydrogen bonds to form between atoms in the backbone. In a couple of cases here, you can see a hydrogen bond form between this electronegative oxygen here and this partially positive hydrogen here that is bound to that nitrogen right there, oxygen and hydrogen, oxygen and hydrogen. So what you're seeing here is all of the hydrogen bonds are formed between atoms in the backbone, and again, not between any atoms that are in the side chains. The side chains are not involved here. In an alpha helix, we find about 3.6 amino acids per complete turn of the helix, and hydrogen bonds will typically occur between atoms that are 4 amino acid positions away from one another. If we move on to the beta sheet, this consists of linear stretches of polypeptide that then bend backwards and orient themselves parallel or anti-parallel to one another. So even though this structure looks very different compared to the alpha helix, it is still very similar in the sense that hydrogen bonds are the essence of what is being used to hold these structures together. Now, as far as secondary structures go, because these are localized structures, you are typically going to find several examples of each within the fully folded structure of any given protein. So there's no rule that says that you can only find alpha helices or only find beta sheets or even one of each in a fully folded polypeptide. So a single polypeptide can and often will have multiple secondary structures formed throughout the entire length of the backbone. In this illustration here, we can find the beginning of the protein here, which is the N-terminus. We can see quickly we form an alpha helix here, then a couple of beta sheets, then another alpha helix, another set of beta sheets, and then 2 more alpha helices before we eventually reach the C-terminus or end of the protein here. So the first thing we want to remember is that these structures are held together with hydrogen bonding. And then the next thing we want to be aware of is that even after these secondary structures have formed, the protein is not fully folded yet. Secondary structure is just a very localized structure. If we're going to fully fold the structure, we're going to have to get interactions between amino acids in the protein that are far away from each other rather than just close by each other. And then finally, you want to make sure you are aware of how the side chains protrude from these secondary structures. In the case of the alpha helices, they tend to stick out at a 90-degree angle, so they're just going to stick straight out like I'm kind of indicating with my mouse here. And then similarly, they protrude out in a similar way from the beta sheets. This means, again, that the side chains do not participate in secondary structure, but the way in which they protrude from the secondary structures is going to be very important for when we move on to tertiary structure. So the next level of structure, which is tertiary structure, is going to be based on how the side chains interact with each other from far away. So moving on to tertiary structure now, if secondary structures are formed because of the attractions between amino acids that are close together, then tertiary structure is formed because of attractions that occur between amino acids that are actually very farly positioned away from each other in the polypeptide. However, there are a couple of important distinctions we need to make here to separate tertiary structure from secondary structure. Tertiary structure is based on attractions between side chains rather than atoms in the backbone. Number two, tertiary structure is based on attractions between amino acids that are far apart in the primary sequence. And number three, tertiary structure can use hydrogen bonds, but it can also utilize other attractive forces other than hydrogen bonds, as we will see here in just a moment. So before we start discussing the attractive forces that hold a protein's tertiary structure together, let's talk one more time about the classes of amino acid side chains that we talked about in the previous video. You have the nonpolar or hydrophobic amino acid side chains, and we'll go ahead and lump the aromatic ones in here as well. You have the polar, hydrophilic, but not completely electrically charged amino acid side chains. And then you have the acidic side chains, which are negatively charged, and the basic side chains, which are positively charged. So for each of those different types of amino acid side chains, when we're determining what the optimal tertiary structure is going to be, we want to ask the question, what kind of interaction would that side chain most like to engage in? So to answer that question, we'll go ahead and start with the hydrophobic side chains. Remember, the word hydrophobic literally means water-fearing. So the hydrophobic side chains, because they are carbon-hydrogen heavy and because they are nonpolar, they are not going to interact well with water. The thing to also remember is that a freely soluble protein will be almost completely surrounded by water. So as the protein folds, the backbone is going to bend and twist to position all of the side chains strategically. So as it pertains to the hydrophobic side chains, our question is, would the hydrophobic side chains rather point outward towards the water or inwards and be hidden away from the water? The answer, of course, is that hydrophobic side chains will prefer to be buried in the protein's interior so that they will be hidden away from the water that they do not interact well with. Similarly, let's look at what this means for hydrophilic side chains, like threonine and serine and glutamine. So for hydrophilic side chains, again, we're talking about a soluble protein that is surrounded by water. The hydrophilic side chains are going to want to stick towards the exterior of the protein so that they can point towards the water that is in the solution or the bodily fluid solvent. So these hydrophilic side chains will be very apt at making hydrogen bonds. So these will form hydrogen bonds not only with each other, but with the water that surrounds the protein. And this is by and large going to contribute to the solubility of the protein in a given bodily fluid. So again, the hydrogen bonds formed between the water in the solvent and these side chains will help keep the protein soluble in bodily fluids and keep it from falling out of solution. And then hydrogen bonding can also be used to help to maintain structures within the protein. So as far as tertiary structure goes, it's not necessarily just these hydrophilic side chains pointed out towards the water. These side chains can also hydrogen bond with one another to help to hold structures together, very similar to what we saw with the secondary structures. As far as the basic and acidic side chains go, I mentioned in the previous video, these fully electrically charged side chains can interact with water, but what they're probably going to prefer to do is form ionic bonds with one another. So these charged side chains can interact with each other, the negative on the acidic side chains and the positive on the basic side chains, and these types of ionic bonds that form between them are called salt bridges. So we've seen in a previous video that these salt bridge attractions are very dependent on a stable and unchanging pH value, and if a given pH value gets too extreme, either too acidic or too basic, these electrical charges can disappear, and that can take away this very important attractive force that is holding a protein together, and we run the risk of a protein denaturing if the pH changes too much. Another type of intramolecular attraction we have not talked about is called a disulfide bond. Disulfide bonds are mediated entirely by one very particular amino acid, and that amino acid is cysteine. Cysteine is one of two amino acids that contain sulfur, the other being methionine. So these sulfhydryl functional groups that you see right here, this SH, are capable of interacting with one another, and if redox conditions in the cells are allowing for it, they can form a disulfide bond, which is a type of strong covalent bond that will help to hold this tertiary structure in place. Since these are covalent bonds, these disulfide bonds are much stronger than, say, hydrogen bonds or ionic bonds, and thus they are going to be harder to break, and they will contribute more to the stability of the folded structure of a given protein. And here in this illustration, you can actually see all of the different types of intramolecular attractions that contribute to a protein's tertiary structure. Not only the salt bridges or ionic bonds that you see here, the hydrogen bonds that you see right here, the disulfide bond that you see here with two cysteines, but also what are called hydrophobic interactions, in which those hydrophobic side chains bury themselves at the interior of the protein. They can interact well together because they have very similar nonpolar chemical properties. Okay, so now we will move on to quaternary structure. Quaternary structure will only apply when you have two or more separate folded polypeptides that assemble into a complex. On the left side of the screen, you can see separate folded polypeptides that for now are all kept separate from one another, but if they start becoming attracted to one another and they form a large multi-subunit complex, this is what is referred to as quaternary structure. If a given protein in the cell consists of more than one single subunit, we would say that that protein has quaternary structure. Proteins that consist of just one single folded polypeptide and nothing more than that never get above tertiary structure. So quaternary structures are going to be held together by the same sorts of intermolecular forces that we saw in tertiary structure, particularly hydrophobic interactions and disulfide bonds. So let's look at two examples of proteins that are held together by quaternary structure. The first we'll introduce is called an antibody. Antibodies are quaternary proteins that are produced by cells in the immune system, and they help to aid the body in cell recognition and bodily defense against invading pathogens like bacteria and viruses. These antibodies, which are also called immunoglobulins, the one you're looking at here is called immunoglobulin G, or IgG. These consist of a total of 4 separate folded polypeptide subunits that are held together by disulfide bonds. These proteins consist of 2 of what are called heavy chains, which are the blue subunits here, and 2 light chains, which are the purple subunits here. The two heavy chains are held together with disulfide bonds, and each heavy chain is attached to a light chain by disulfide bonds as well. The next example we will look at is hemoglobin. Hemoglobin, we understand so far, is an oxygen-carrying protein that is found in red blood cells, but it is also a quaternary complex that consists of 4 polypeptides in total, 2 alpha globin subunits and 2 beta globin subunits. This quaternary complex, again, contains 2 identical alpha globin subunits, 2 identical beta globin subunits, and each subunit contains a heme prosthetic group. A prosthetic group, which you can see for this case, this heme prosthetic group is illustrated here. A prosthetic group in a protein basically serves the same function that, say, a prosthetic arm would serve someone who has lost their natural arm in an accident. So for someone who has lost their arm, they no longer have the ability to use that arm because it is no longer there, but if they attach a prosthetic arm, that prosthetic arm is not a natural part of their body, but serves some sort of purpose and gives their body some sort of function it did not have before. In the case of proteins, prosthetic groups are not a natural amino acid-based part of the protein, but they impart to the protein some sort of function that it did not have before. In the case of the heme prosthetic group, it allows hemoglobin and the globin subunits to bind to and carry oxygen. Again, prosthetic groups bind to proteins and give the proteins some sort of function that cannot be performed by the amino acids in the protein. So again, if you pay attention to the heme prosthetic group here, it consists of an organic porphyrin ring that coordinates a single iron atom at its center, and this iron atom has a high affinity for oxygen atoms, and this accounts for both hemoglobin and myoglobin's ability to carry oxygen around. Speaking of myoglobin, myoglobin is a very similar protein to hemoglobin. It is an oxygen-carrying protein, except myoglobin is found in skeletal and cardiac muscle rather than in red blood cells. If you compare the tertiary structures of myoglobin and beta globin, then you would find that they are almost identical to one another. The difference, though, is that myoglobin does not form quaternary structure. The natural state of myoglobin in muscle fibers is to be a single polypeptide that does not form quaternary structure. So this is very interesting because if we compare the oxygen binding curve of myoglobin to hemoglobin, for myoglobin you can see that myoglobin has a very high affinity for oxygen because its curve is pushed almost entirely to the left. So we can see here that at even very low levels of free oxygen, almost 100% of myoglobin will be bound to oxygen. The only way to get myoglobin to let go of its oxygen is for free oxygen levels in the cell to reach near zero. So in this regard, myoglobin acts kind of like an oxygen battery so that it will release its store of oxygen only when muscle fibers start to critically run low on oxygen levels. So, like I said, this manner of binding and releasing oxygen in such an extreme fashion works for muscle tissue, but it cannot possibly work like this in the blood, where hemoglobin needs to be able to bind to oxygen and let go of it in the appropriate situations. So if we compare the oxygen binding curve of myoglobin to that of hemoglobin, you can see these curves look very different. For hemoglobin is very similar to myoglobin in that it has a very high affinity for oxygen when free oxygen is high, like it is in the lungs, but hemoglobin's affinity for oxygen is much lower when free oxygen is on the lower end, like it is in the tissue. So what this allows hemoglobin to do is when it passes through the pulmonary capillaries in the lung, it binds to oxygen with high affinity and can now carry it throughout the body. But when hemoglobin reaches the systemic tissue capillaries where oxygen is much lower, hemoglobin can let go of oxygen and efficiently deliver it to cells that are actively going through cellular respiration. So if you're wanting to know, well, if myoglobin and the globin subunits of hemoglobin are so similar at the tertiary level, why do they have such different behaviors as it pertains to oxygen transport? So again, basically we answer the question there. Since they do have such near identical tertiary structures, the difference here has to have something to do with the fact that hemoglobin forms quaternary structure and myoglobin does not. So there has to be some sort of subunit communication in hemoglobin that is not possible with myoglobin. That will be something that we discuss much later in the semester, but for now, all you need to appreciate is that the fact that myoglobin and hemoglobin are so different in terms of function, it has to do with the fact that myoglobin does not form quaternary structure and hemoglobin does. Okay, that's going to wrap up this video. Here's a list of vocabulary terms you probably ought to be aware of. And for checking your understanding, number 1, what important information can we get from a protein's primary structure? Number 2, what kinds of chemical bonds are used to stabilize each of those structures? And number 3, what accounts for the differences in oxygen binding observed for hemoglobin and myoglobin? So that's going to do it for this video. Thanks for your attention. If you have any questions, go ahead and drop them in the comments section on YouTube. Otherwise, I will see you for the next video.